All right, with that, it is my pleasure to introduce our first speaker, Dr. Ineke Brouwer. Ineke did her PhD in Amsterdam with Drs. Wuta and Peterman, where she used optical tweezers to understand membrane function and DNA repair. She is currently a postdoc with Dr. Lenstra, where she studies chromatin remodeling during transcription. So today, Ineke is going to tell us about her postdoc work, but she is hoping to move on soon and is on the job market. So if you know of a position that would be a great fit for Ineke, or if you want her to join your department, please reach out to her. So I won't take any more of your precious time, Ineke, and I'll just hand it over to you to tell us about your work.
Thank you for the kind introduction. Indeed, I will tell you about my postdoc work, which focuses on understanding the link between chromatin remodeling and transcription dynamics. We recently posted this story as a preprint on bioRxiv, so please check that out as well. To start transcription of a gene, several sequential steps have to take place. First, a transcription factor binds to its binding site in the gene's promoter. This then recruits Tata binding protein, or TBP, which binds to a Tata or Tata-like sequence close to the transcription start site. This binding of TBP is the first step in recruitment of the pre-initiation complex, which is a large protein complex entailing all of the transcription machinery. Once the PIC is assembled, polymerases can start to elongate and RNAs can be produced. Now we know that transcription occurs in bursts. These are short periods of time interspersed by, in which a few RNAs are produced, interspersed by periods of no transcriptional activity. We know that this bursting behavior is a major contributor to cellular heterogeneity and cell-to-cell variability, but the underlying mechanisms that control transcription and bursting remain largely unknown. Another important aspect in regulating transcription are nucleosomes. By wrapping the DNA, nucleosomes control the accessibility of specific promoter elements, such as the binding sites of transcription factors. So the question that I want to address here is what the link is between the remodeling of these promoter nucleosomes and the dynamics of transcriptional bursting. And to address this, we use the GAL10 gene of budding yeast as a model system. This gene is only expressed in galactose-rich media, so we can induce the system just by addition of galactose. Now, the first step in answering this question is to determine what the chromatin structure is of the GAL10 promoter in both raffinose, where the gene is inactive, and in galactose, where the gene is active. For this, I've used MNA-seq, a method where all the DNA that's not bound by a nucleosome is digested. Subsequent sequencing of the remaining DNA then reveals where any nucleosomes were bound. And to discriminate between the canonical stable nucleosomes and the more loosely bound, partially unwrapped fragile nucleosomes, I've used two different concentrations of the MNase enzyme. And this allows to map these two types of nucleosomes separately. Now, in inactive conditions, the promoter region contains 6 nucleosomes, of which one nucleosome, which is located directly over the GAL4 binding site. And this nucleosome is predominantly a fragile nucleosome. Upon addition with galactose, 3 of these nucleosomes in the promoter, including the nucleosome that was directly over the Tata element, are evicted by chromatin remodeling complexes. At the same time, the nucleosome close to the transcription start site moves slightly upstream. But the fragile nucleosome over the GAL4 binding site remains present even in active conditions. So schematically, the promoter contains 3 nucleosomes, of which the central one is a fragile nucleosome in active conditions. Now, because the GAL10 promoter is shared with the GAL1 gene, we assume that the 3 nucleosomes closest to GAL10 are most important for regulating GAL10 transcription. So for today, I'll focus on these 3 nucleosomes in the promoter region. Now, to understand the effect of the remodeling of these nucleosomes on transcription dynamics, I target the protein complex RISC. This is a chromatin remodeling complex known to be responsible for positioning of promoter nucleosomes specifically. Using a conditional depletion system, I measure both the nucleosome positioning in the promoter region in cells with and without RISC, and subsequently, I measure the effect of this changed promoter structure on transcription dynamics. To measure how the promoter nucleosomes change when we deplete RISC, we again performed MNA-seq in cells with and without RISC. And what we observe is a similar pattern for the stable nucleosomes, with perhaps only a slight increase in a nucleosome density close to the transcription start site. A more striking difference is observed if we look at the fragile nucleosomes. Here, the coverage is much lower, specifically around the binding site of the transcription factor Gal4 and also around the transcription start site. So this schematic, which will come back at many places throughout my talk, indicates which nucleosomes are predominantly affected. To measure what the effect of this change promoter architecture is on transcription dynamics, we use the PP7 system. Here, a sequence encoding for the PP7 stem loops is inserted at the 5' end of Gal10. Upon transcription, the RNA then contains these stem loops, which are instantaneously bound by a fluorescently tagged code protein that we've expressed in the same cell. This code protein then accumulates at the transcription start site, and we can use it to monitor the transcription dynamics of an individual gene in real time in an individual cell. From such movies, we can extract intensity time traces, and by using a thresholding approach, we can determine all the individual parameters of transcriptional bursting. So the duration of a transcriptional burst, the time between consecutive bursts, the intensity during a burst, and also the induction time, which is the time between the first addition of galactose and the first burst of transcription. Now, of course, we don't do this in a single cell, but rather we do this in a group of cells with risk and a group of cells without risk. And I represent the data here as a heat map, where each line is an individual cell, and in yellow, you see the transcription site intensity as it is bursting over time. Now, from these heat maps, we can measure the distributions of all of these bursting parameters and compare them between cells with and without risk. So in this plot, each individual dot is an individual cell where we've determined its induction time. And what we find is that cells induce a little bit slower in the absence of risk. And also the time between consecutive bursts is slightly increased if cells are lacking risk, while the other parameters of transcriptional bursting remain unchanged. So together, this means that each burst of Gal10 transcription is slightly delayed in cells that are lacking risk. So we conclude that the fragile nucleosomes over the Gal4 binding sites and the transcription start sites are important for regulating the frequency of bursting. Now we know that risk is not the only remodeler of promoter nucleosomes. So we also looked at the effects of depletion of SWI-SNF, which is the other important remodeler of promoter nucleosomes. When we performed MNA-seq in cells with and without SWI-SNF, we observe a changed configuration of the stable nucleosomes with increased nucleosome density over the Tata element. And if we deplete both RISC and SWI-SNF simultaneously, we see an even larger increase in this nucleosome density, as indicated here by the white arrow. From that, we can conclude that RISC and SWI-SNF together regulate all the three nucleosomes in the Gal10 promoter region. Now, we also looked at the effects on transcription dynamics. And to assess the combined effect of multiple perturbations, we devised an analysis method, which we've termed dynamic epistasis analysis. In this way, we measure the effect of our perturbations on each dynamic parameter of transcriptional bursting. So for this, we plot the fold change in each individual parameter upon a specific depletion. So as I showed before, RISC depletion leads to increased induction time and increased time between transcriptional bursts. Doing the same experiment with SWI-SNF, we show a delayed induction time, but no change in steady state transcription. From these individual fold changes, we can calculate what the expected effect of a double depletion would be on each parameter if we assume that RISC and SWI-SNF act independently. I show this range in gray in these plots. A larger than expected effect would indicate that these processes are redundant or act within the same pathway. A larger than expected effect would mean that the processes are redundant, while a smaller than expected effect would indicate that they are antagonistic or within the same pathway. Using our double depletion cells, we can then measure what the combined effect is, and we observe a larger than expected effect for both induction time and the time between bursts. So that shows that RISC and SWI-SNF redundantly remodel the promoter nucleosomes and regulate transcription dynamics. Now, surprisingly, we still see quite a large amount of transcription in these cells that are lacking both RISC and SWI-SNF, so are essentially lacking promoter nucleosome remodeling in general. So our next question was what the mechanisms are that control the regulation of bursting by nucleosome remodeling. And for this, I want to look at remodeling of two of these nucleosomes separately. First, I want to show you what is going on at the fragile nucleosome around the Gal4 binding sites. And then I want to talk about what is going on in the nucleosome covering the Tata element. So let's start with the region around the Gal4 UAS. To understand what is happening here, we've looked at cells where we've lowered the protein concentration of Gal4 by deletion of one of the two copies of the Gal4 gene, and we've combined this with risk depletion. What we observe is that just lowering the levels of the transcription factor Gal4 increases the induction time, but doesn't change the time between consecutive bursts. If on top of that, we also deplete risk, we observe the expected effect in terms of induction time, but a synergistic increase in the time between bursts. So that tells us that risk remodeling and Gal4 binding act redundantly and are thus important for regulating each burst of Gal10 transcription. Now, to understand what is going on at the nucleosome around the Tata element, we wanted to do the same experiment, but now with TBP instead of Gal4. So we looked at cells where we've lowered the protein level of TBP. Now, just reducing the TBP levels perhaps decreases the induction time slightly, while the time between bursts is again unaffected. If on top of that, we also deplete risk, we see in contrast to what we observed with Gal4, we now see an synergistic increase in the induction time, but the expected effect in terms of the time between bursts. So that tells us that risk remodeling of this Tata nucleosome is important for TBP binding and to allow the first burst of Gal10 transcription to occur, whereas remodeling of the nucleosome around the Gal4 binding sites is important for each consecutive bursts. Now, the redundancy between TBP and risk that we observe suggests to us that maybe in the absence of risk, TBP is somehow able to take over risk's function in removing this Tata nucleosome. So to confirm this, we've performed MNA-seq in cells with where we've depleted risk and reduced the levels of TBP. Here, we indeed find increased nucleosome density over the Tata element if we compare this to cells where we just depleted risk. Now, this indeed confirms that TBP itself contributes to the nucleosome positioning in these risk-depleted cells. So we interpret this as follows, where in wild-type situation, risk is responsible for removing the nucleosome over the Tata element, such that TBP can bind. When risk is lacking, there's more nucleosome density at the Tata, but TBP can still bind to this nucleosomal DNA and then push away the nucleosome from the Tata to allow transcription to activate. However, if we've also reduced the amount of TBP in the cell, this activity is more difficult and thus takes longer, resulting in an increased nucleosome density and thus a synergistic downregulation of the activation of the transcription. Once, however, TBP is bound and it has pushed this nucleosome away, it keeps the Tata free of nucleosomes and subsequent bursts are not affected, which explains why we see no effect in the steady state transcription. Now, we hypothesized that this ability of TBP to displace this particular nucleosome may depend on its dwell time of TBP at the Tata element. So only sufficiently long and stable binding of TBP might facilitate this nucleosome removal. So to test this, we've mutated the Tata element of Gal10 to reduce the TBP dwell time. What we expect is that in the absence of risk, TBP will no longer be able to remodel any nucleosome in the Tata region and thus will not allow for Gal10 transcription to initiate. When we performed live cell imaging experiments in these Tata mutant cells with and without risk, we indeed observe that in the absence of risk, part of the cells can no longer activate Gal10 transcription. And this is due to the reduced remodeling capability of TBP. And if we look at the cells that do activate, we find that these are able to transcribe Gal10 in a risk-independent manner. So from this, we conclude that it depends on the initial stochastic positioning of the nucleosome around the Tata element, so that if this nucleosome covers the Tata element, in the absence of risk, this nucleosome cannot be remodeled because TBP doesn't bind stably enough to compete with this nucleosome. If, however, there happened to be no nucleosome over the Tata element, remodeling is not needed, and transcription can thus occur independently of risk. Now, to strengthen this link between the dwell time of TBP and its remodeling capabilities further, we also wanted to increase TBP's dwell time. For this, we used a depletion of the enzyme MOT1, which is the enzyme that's responsible for removal of TBP from the Tata element. We expect that increasing the TBP stability in this way would increase the ability to actively remodel the nucleosome at the Tata. Doing live cell imaging experiments in these cells depleted of MOT1, we see no effect on the transcriptional activation. However, if on top of that we also deplete RISC, we observe a smaller than expected effect, and even a smaller effect than just depletion of RISC. So that means that MOT1 depletion rescues the effect of RISC depletion, and indeed remodeling by the stabilized TBP alone is enough to efficiently keep the promoter in an active state. And this is independent of RISC. Now, taken together, I've used a unique analysis of single and double perturbations to elucidate how the different promoter nucleosomes have specialized functions in regulating the different parameters of transcription dynamics. On the one hand, continuous remodeling of the nucleosome around the GAL4 binding site is needed to allow GAL4 to bind and to allow the start of each transcriptional burst. In addition, the first burst of transcription is controlled by remodeling of the nucleosome around the Tata element in an interplay of remodelers and TBP. Here, we've revealed a novel role for TBP in competing with this nucleosome as long as the dwell time of TBP on DNA is long enough. And together, these results contribute to a better understanding of transcriptional bursting and thus the mechanisms controlling cellular heterogeneity. So with that, I would like to end by thanking everybody involved in this project. Of course, the members of the Lenstra Lab, Fred van Leeuwen, and also the core facilities at the NKI. So we've currently put this story out on bioRxiv. And if you want to know more, I'm very happy to answer any questions now or later in the coffee chat, or you can always drop me an email. Thank you.
Thank you, Ineke. That was really nice. I will remind the audience that you can either post your questions in the Q&A or you can raise your hand and we'll unmute you so you can ask your question. And like Ineke already mentioned, there will be a coffee chat after this. So if we don't get to all the questions or you have more in-depth questions, feel free to leave us a message in the chat that you want to stay and we'll promote you to a panelist at the end of the talks. All right, so I'll start with a question that came in the Q&A from Hannah Richter. She says, great talk. I noticed that in the risk TBP depletion condition, it wasn't just the Tata nucleosome affected, but also increased occupancy at the GATA UAS. Any comments on possible mechanism of this?
Yes, so let me try to go back to that plot, if it will let me. So I think you're talking about this increase over here. So I think what you see here is a signature of the fragile nucleosome over the GAL4 UAS that is always that is also present if you look at the fragile nucleosome distribution. So we think that this difference is probably because the samples are digested a little bit different between the two conditions. So we think that this is not a real effect, but because of the variation between the experiments. So that this is actually a fragile nucleosome and not a stable nucleosome. So this is not really a biological difference between the two conditions.
Okay, we have a bunch of raised hands. So I'll start by allowing Arvind Pandey to talk.
Hi, can you hear me?
Yes.
Mm-hmm.
Yeah, very nice talk. I have a very kind of a basic question. So you mentioned two types of nucleosome, stable nucleosome and fragile nucleosome. So how these two nucleosomes are different? Are they different in terms of DNA binding affinity or positioning? And my second question is how these nucleosome affects the remodeler activity in general.
I think there's a lot of debate and discussion in literature about what exactly these fragile nucleosomes are and what the difference between these and the more canonical stable nucleosomes are. So for the purpose of our study, we've just classified the two types by a difference in their ability to be digested by MNAs. And at least for the fragile nucleosome over the GAL4 binding sites, it is known from previous studies that there is really a nucleosome there and that there are histones binding there. So it seems to be a nucleosome that contains all of the nucleosome components. But exactly what the difference is, I think, is still unclear. I always have interpreted it as something that is probably a bit more loosely bound and also more dynamic, so easier to remodel. And in terms of remodelers, it is known that different remodelers act differently on fragile or stable nucleosomes. For example, already from my data, I've shown that the fragile nucleosomes are more affected by risk, while the stable nucleosomes are less affected by, well, at least at the GAL10 locus, is less affected by risk, but more by SWI-SNF. And also genome-wide, you see less effects on the stable nucleosomes by SWI-SNF, but more by risk. So I hope that answers the question.
Yeah, I got it. Just want to confirm, like you said, fragile nucleosome loosely bound and a stable nucleosome kind of more tightly bound, right?
Yes.
Okay, got it. Thank you.
You're welcome.
Okay, we'll do two more questions. So I'll start with David Arnosti's question, which says, please explain your model for how reduced levels of TBP lead to an accelerated induction rather than delayed. They're expecting that they say that this is unexpected.
Yes, I agree. So I think you're talking about this effect, so that if we just lower the levels of TBP without even touching the remodelers, there seems to be a slightly faster induction of GAL10. So we've done this experiment in many cells, and the difference doesn't seem, well, it's only borderline significant. So here we think that this is an artifact of the tagging of TBP that we had to do to be able to deplete part of the TBP population, and isn't really a biological effect, because I agree that if you would expect that if you have less TBP, you wouldn't expect things to happen faster, but rather slower.
Okay, and we'll take one last question from Jason Fan. There's lots of other questions, so please stay for the coffee chat or email Ineke with your questions. All right, so Jason Fan's question, he starts by saying, hi, great talk. I wonder whether double depletion of RSC and SWI-SNF would totally negate TBP's ability to displace nucleosomes, since the nucleosome won't be able to be removed from the TBP binding site.
Yes, I wonder that too. So I think right now, even in cells where we removed part of TBP and removed RSC completely, we still in the end see some transcription. So it would indeed be interesting to then also deplete SWI-SNF and then see if transcription completely stops. But that's an experiment that we haven't done at the moment.
Okay, great. So again, lots of questions. Please let us know if you want to stay for the coffee chat, and we'll promote you later, but I'll hand it over to Christine.